think we have to get used to this idea that in the online world, you could be anywhere in the world and it could be any time of day for you as well. So I'm very happy that you're able to join us for our weekly seminar. And we're delighted today as our speaker to welcome Dr. Morgan Craig from Université de Montréal. And she will speak about identifying mechanisms of immunopathology in severe COVID-19 in a realistic virtual patient cohort. Morgan, please.
Thanks. I'm just going to share my screen and get everything set up. Great. And then I'm just going to turn off my video while I present, and then I'll turn it back on for questions. So thanks everyone for the invite and to the Fields Institute and Jang Hong and everyone there, because I'm very happy to be with you. I think that I might be taking a bit of a different focus today because I'll be looking at the within host model of COVID-19 and some of the work that we've been doing to try and understand the immune response. So looking at within host models and what this means, implications for potential therapies. So my lab is focused on using mathematical and computational models of diseases to understand the mechanisms of pathologies writ large, and also to improve and optimize therapeutic approaches. To study novel treatments in both preclinical, we do a lot of preclinical work, and also clinical applications. We use our in silico virtual trial approach that we've developed, but which is also something that is used in the pharmaceutical industry a lot, for example. So in this framework, we choose a disease, we develop a mathematical model of the disease, which is constructed from the known pathologies, but also known mechanisms of the system of interest. And from there, we can generate a cohort of virtual patients in which, as you can see here, the cohorts are separated out, but are the same virtual patients in the same in different cohorts. And in this way, we can look at different therapeutic options, different characteristics, considerations, I guess you could say, to be able to dissect and understand mechanisms that lead to response or non-response to treatment, and also to reveal the causal drivers of disease and the mechanisms of these therapeutic responses. So as part of this work, we've been involved in multiple studies of within host responses in infectious diseases. And for example, during my postdoc with Allison Hill at Harvard, who's now at Johns Hopkins, we focused on the preclinical development of a novel gastric retentive extended release device that could be used for PrEP, so for pre-exposure prophylaxis, and also for anti-HIV therapy. And for this, we took into account the resistance mutations, which are kind of indicated here, that may occur during therapy. And this model is kind of a precursor to some of the work that we've been doing in SARS-CoV-2 infections, where we're looking at how the immune response, how the within host response evolves, but also how the virus may evolve and how that changes the immunological response and disease. So the work I'm going to talk about today was really accomplished. I mean, it's not, obviously it's never just one person's work. This is a collaboration that grew out of another collaboration that we work on with Paul Macklin at Indiana University, who's leading a model of the tissue level response to SARS-CoV-2. So we're a group of mathematical biologists, experimentalists, and immunologists who have been introduced as a part of this tissue level model and who work now together on the model I'm going to talk about today. And just to highlight Adrienne Jenner, who it says Université de Montréal, but now she's a lecturer at Queensland University of Technology. She was a postdoc in my lab. This is mostly her work, and this was accompanied with two other undergraduates in my lab, Sophia and Viv Crow. Oops, sorry. as I mentioned, we're working on a tissue level response model. So this is really in the micro level. And this is done as part of a large international consortium working on building a computational framework for this tissue level model. And if you're interested, I've left the link there, and they're also available on my website, or if you just search Paul Macklin COVID to get more details of that model, which is based on an agent-based modeling framework. But the work I'm going to talk about today is more at the systemic level. And here we've really been focused on delineating the pathological mechanisms leading to severe COVID-19. And that will be my focus today. And as you can see here, I'm not going to click on it, obviously, but we have a preprint out. This is a paper that is under review. And at the end, I'll talk a little bit about some of the work that is ongoing. So within the host, changes in viral load can be described by what's called the basic viral dynamics model. And if you're familiar with an SIR model, you're likely familiar with a viral dynamics model. They act largely the same. In this model, you have, instead of susceptible individuals, susceptible cells that become infected through contact with viral particles or virions, here in my very idealized virus figure. And these infected cells will eventually die. And during the process of infection, they're releasing additional viral particles, and also through death, they may release viral particles. And these released virions then go on to infect other cells, which continue the viral replication cycle within the system. So in this way, it's similar to an SIR model in the sense, or I guess an SIE model expired, where you have, or D, where you have individuals who are, coming into contact with others and infecting them. Here, you're looking at the virus particles that are infecting other susceptible cells. What this basic viral dynamics model kind of smooths over is the contribution from the immune system to these processes. So in viral infections, both the innate and the adaptive immune systems are activated to clean up virus and kill and remove infected cells. And in an aberrant immune responses, when you have dysregulation or things that aren't going right, the adaptive immune system may not be able to mount an effective response. And this, in viral infections in particular, can lead to an increased number of innate immune cells and a large amount of dysregulation in the system. And this is what we've been talking about when we hear in COVID-19 about cytokine storm. So it's hyperinflammation and dysregulation of the translation from the innate to the adaptive immune response. And in these cases, as I mentioned, which is typical of severe COVID-19, this dysregulation can cause damage to uninfected, susceptible cells. And this then aggravates the process because now you also have an immune response that is responding to damage in addition to the viral infection that's ongoing in the system. And so just to give a bit of biology before we go into the model, I'm just going to talk you through or walk you through some of the key facets and parts of the immune response to SARS-CoV-2. So this immune response begins with the secretion of type 1 interferons. So the cell will become infected and it secrete type 1 interferons, which signal to act to shut down viral replication within the cell, and also to signal to neighboring cells to do the same. And so in this way, type 1 interferon acts to kind of tamp down the viral replication process and hopefully salvage essentially nearby tissue. After this interferon signaling starts, a set of cytokine signaling cascades is set off, and you can see them here in the bottom. I've highlighted the ones that I want you to pay attention to because they're the ones that we've modeled, and I'm going to talk about in a second. And these signaling signals work to recruit various immune cell subsets. And so in the healthy immune response, if your body is acting appropriately, alveolar or resident macrophages within the tissue, and here we're talking and thinking specifically about lung cells, but of course this is happening in wherever there may be viral infection. Sorry? clear, sorry, so yeah, if you have questions, please just let me know. So yes, so macrophages are there, they're in the tissues and they're immediately able to respond. And they do this by apoptosing infected cells or by phagocytosing infected cells. And there's also these signals for CD8 T cells through the signaling cascades put out by infected cells that are recruited to the site of infection and then can apoptose the infected cells. And so because you have this dual effect, you have clearance from the alveolar macrophages, you have a certain amount of inflammatory macs that can come from the resident tissue macrophages, and you have the CD8 T cell response, you have a rapid clearance of the infection and minimal tissue damage. And so, you know, this is the kind of, you know, largely asymptomatic or very mild infection dynamics that you would see in somebody with a well-mounted immune response to the infection. However, in dysfunctional immune responses, both the CD4 positive and CD8 T cells are not efficiently activated. So they're not arriving at the right time, in the right number, primed in the right way. And the innate response, the macrophages that I mentioned earlier, monocytes, which are a type of myeloid cell, and even neutrophils, which are not typically known to be very active in viral infections, but which will come in to help with clearance of virus. Particularly in the case where the adaptive immune system hasn't taken over. So these innate immune cell subsets will kind of start controlling or attempt to make up for the lack of innate adaptive immune response, and can cause hyperinflammation, the cytokine storm that I mentioned earlier, and lots of tissue damage. And so this hyperinflammation and dysfunctional immune response are characteristic of severe COVID-19. Here are some concentrations of IL-6, which is a type of interleukin that's involved in T cell response, and also concentrations of T cells from patients with mild in blue and severe in red patients with COVID-19. And in particular, patients with severe COVID-19, as you can see in the slides on the left, exhibit elevated IL-6 concentrations, lymphopenia, which is a lack of T cells, and you can see that their T cells are depressed, and a delayed interferon response. And this is particularly interesting because this is replicated in both the original SARS strain and in MERS. And delayed interferon appears to be related to the inability of the adaptive immune system to mount an effective response. And so this has been replicated or seen, as I mentioned, in other beta coronaviruses. It's also an emerging component of the SARS-CoV-2 infections. And we had previously developed many different models of different disease processes. And based on our previous work, we sought to develop a mathematical framework in which we could probe the mechanisms that were regulating this immunopathology in COVID-19. So what we did to start was to adapt the basic viral dynamics model that I had shown you earlier, first to basically parameterize it to understand early infection dynamics. So though I'm showing the immune response here, for the moment, we're not taking into account any immune response, but we use what's called the target cell-limited viral dynamics model, in where we account for what's called the eclipse phase. So this is the time after cells are infected, but before they begin producing virus. And to do this, we use delay differential equations, but you can also add a compartment for the eclipse phase cells. And you'll note that we also are using a logistic growth function because we predict or model, hypothesize that eventually your lung cells, unless obviously there's enough damage to kill you, there will be rebound in your lung cells after the infection clears. And so we fit this model to data from macaques. I'm sure you've seen this data before. It's from Munster et al. And they inoculated macaques in a very controlled way. Because we were really focused on getting the early infection dynamics right, and this is not something that you'll get from human patients, this kind of data, we fit to the macaque data. And as you can see, I'm showing here is the fit to the viral load, but then also, as I mentioned, this rebound in the number of susceptible cells over time. But we can also fit human data with this model. So it's essentially a step towards getting the immune or the infection dynamics correct in humans, and to fit these human patients, which were from Singapore, if they're indicated by S, or G, if they're from Germany, we were able to fit them by fixing most parameters in our model from the macaque model, and refitting just the initial viral load, which is no longer constant as it was in the macaque data because that was controlled inoculation. The time of infection, initial infection, since patients are typically not identified as PCR positive until days after becoming infected, and the viral clearance rate. And I guess just to say that in the next few slides that I'll show you, we're going to be modulating this viral clearance and this also the clearance of infected cells by adding in the immune system. So I'm not too concerned that we are refitting a viral clearance parameter.
Okay, I'm sorry, I'm using my role as a co-host. What about the delay? Is that the same for both animals and humans?
Yeah, so we set the delay the same because the delay is inherent to the time for the replication cycle of the virus. So it's a viral specific characteristic. But you're right. So we've worked also on distributed delay models, and this is something that I'd like to look at to see, can we get different, or how accurate are these fits if you put in a distributed delay into that delay.
Thank you.
Great, yeah. Okay, so as I mentioned earlier, after cells become infected, they begin secreting type 1 interferons. And this is again to tamp down the infection process, not just within its own cell, but also in neighboring cells. And so now we're kind of building this model in a piecewise way. So we're going to now add in additional terms to account for the resistant population, what we call resistant. It's more just the population that is now unable to be infected because of this type 1 interferon signaling response. So our model terms throughout were always calibrated to dose response data or other available data. And we typically use stimulatory, so you can see that here, or inhibitory Hill functions. And so what we did was to add in the effect. So there are fewer cells that are able to be infected once the interferon response starts signaling. There's this new population of resistant cells that are not infected at the same rates. There's also now an equation accounting for the clearance or the use of interferon by infected cells and their signaling by infected cells. So again, we're trying to take a piecewise step so that we're not adding everything in at the same at one point and losing kind of the precision, if you could use that word, of what we're trying to model, by adding everything in too quickly. So at this stage, this is what the model looks like. We have this basic viral dynamics model. Virus infects cells, they die. Virus infects susceptible cells, and interferon mediates this by creating resistant cells in the system. And so now we're really going to start to build in, after we had calibrated this, we started to build in the various facets of the innate and the adaptive immune response after infection. So we model the cells that you can see here on the right, and in particular neutrophils, which are typically thought of as more secondary players in viral infections, but which still can play an important role in viral clearance. We modeled them as recruited by G-CSF, which is a cytokine that is that stimulates neutrophils, and IL-6, and capable of inducing death in cells, both infected and susceptible, using NET, so neutrophil extracellular traps. In dysregulated immune response, through the release of reactive oxygen species, neutrophils are known to cause high degrees of tissue damage and to be implicated in hyperinflammatory responses. So we also calibrated the secretion of these reactive oxygen species by the number of neutrophils and account for their damage on susceptible cells and infected cells. We also model, of course, the tissue resident or alveolar macrophages that I mentioned earlier, and their transition into the inflammatory macrophage subtype. These can be derived from the resident macrophages or also from monocytes that are mobilized from the bone marrow. And these are recruited through interferon and GM-CSF signals. These macrophages are really important. They're the ones that are phagocytosing infected cells, and they're helping to clear damage by removing dead cells. And so the inflammatory macrophage response, I mean, as with everything, you don't want too many, but they're really kind of the key players at clearing away infection. And then lastly, we also include CD8 positive T cells. We discount for right now in this model, the CD4 in the, taking antigen into the lymph nodes and CD8s coming out. We just model a delayed process for the arrival of T cells that accounts for kind of these processes happening in the background. CD8 T cells, therefore, are recruited to kill infected cells and apoptose them, as I mentioned earlier, which is enhanced by IFN and mediated by IL-6. And so as you can see, I've been mentioning all of these cell subsets, but we have a number of cytokines that we need to account for in the model. And we do that by modeling them as produced by various immune cell subsets in our model and then used at their target site or their site of action. And for this, we model the pharmacokinetics of each cytokine by explicitly accounting for both bound and unbound concentrations. This is because previously in other work more related to cancer, but also the homeostatic regulation of neutrophils, we found that the cytokine paradigm of bound-unbound being in equilibrium is not upheld at endogenous cytokine concentrations. So we model their kinetics through, or cytokine kinetics through binding to cell surface receptors, internalization, renal clearance, and potential unbinding, though this is not as important for the pharmacokinetics. And for all of the terms in this model, which might seem large, we extensively calibrate against available data. And we use what I call a hierarchical fitting, that might be a term I made up, but I'm not sure, but a hierarchical fitting scheme where we're really isolating subparts of the model, making sure that they can reproduce what is known physiology, and then adding that into the full model. And so here I'm showing, and I won't go into all the details, the kind of fits that we did for data for a variety of different data, for a variety of different considerations that are taken into account in our model. And I just want to, again, emphasize how much this is a team effort and group effort. And really, a lot of this fell to Adrienne and Sophia, who did a lot of the data fitting and integrated in vitro, in vivo, and clinical data. And they obviously did a great amount of work here to digitize all this data and to do a lot of fitting. And we're very happy and confident in our predictions because there was this extensive amount of data fitting. And some of this experimental data actually comes from our own group, including from flu, from Amber Smith's lab, from some mice data. So using our simple viral dynamic submodel, we estimated the parameters from macaques, as I mentioned earlier, and then we integrated all of the data fitting that we had done, the immune cell modeling that I just described, and integrated also a number of clinical studies that characterized the differential immune response in COVID-19. And the idea here was to know whether our model could successfully recapitulate both the mild and severe cases, including with respect to IL-6, interferon, the CD8 T cell response, and also G-CSF concentrations. And so for this, this was not, we did not fit to this clinical data, but the idea was that we had fit to a variety of data sources. And now we want to make sure that our model can accurately or successfully reproduce this clinical data. And in a large part, it does. And so I'm showing here some of the clinical data in dots or in shaded regions in our model predictions, either for mild disease or severe disease. And again, just to reiterate, none of this is fit. This is just comparing to data. So let me just walk you through the results for an average patient using this full systemic model that I just explained. So in each of these graphs, the mild response is the solid line. There are different things on each axis because there's a lot of things to try and summarize here, but I'm going to walk you through it. The severe response is in the dashed line. When we fed this viral dynamic submodel into the full immune response model, we found several characteristics. We found first that in severe disease, there's obviously a reduction in lung tissue during the course of infection in mild and severe cases. This is not surprising. There should be a reduction in lung tissue, mainly because there's hyperinflammation. I'll show you that in a couple slides. We observed, as I mentioned, an increase in all inflammatory immune cell subsets. So here you can see increases in monocytes, macrophages, neutrophils, and also, though importantly, a large and significant reduction in T cells. And as I mentioned earlier, this lymphopenia is particularly observed during severe COVID-19. And then lastly, we found that delays in interferon signals emerged in the average severe case. All of these properties together are characteristic of severe COVID-19 cases. And given that we didn't impose these behaviors, but instead kind of found them as emergent from our modeling, we were confident in our ability to distinguish these two cases. So our model not only acts as, obviously it's built from hypotheses, but there were emerging properties here. We did not build in a reduction in T cells. We didn't build in this delay in interferon responses. And so we really wanted to dig in a little bit further on this and look, this is for an average patient, but obviously, as we know, there's kind of a full spectrum of responses in COVID-19. And we wanted to try and use our model to be able to distinguish, as I mentioned right on the first slide, kind of causal or mechanistic reasons that there may be these observed differences in outcomes. And so for this, we generated, as I mentioned earlier, we used our in silico clinical trial approach, and we generated virtual patients by drawing a subset of their parameters from distributions. And these distributions were informed by experimental and clinical data, some of the data that I showed you earlier. And then we generate these values, we evaluate the model, and then we use simulated annealing to ensure that the trajectories, so predicted trajectories, fell within observed clinical ranges. And this technique was suggested or put forward first by Allen et al., Richard Allen, who's at Pfizer, and we've used it in other adaptations in cancer immunotherapy and other scenarios. And so it's kind of an established technique that we're using in the lab to be able to draw from these distributions and make sure that trajectories fall within ranges. And then once we simulate this, we're getting out the kind of responses that we would anticipate having. And again, this is not like a fixed response, but more a range of responses. We then accept that patient into the cohort. And so in this way, we're really recapitulating the heterogeneity in real patient cohorts, because we're always comparing disease trajectories to those which are seen clinically. So we generated 300 patients in this way. And for each of our 300 virtual patients, we simulated their disease course and compared them using several key metrics. On the top here, you'll see this phi, or psi, sorry, which is what we call an inflammatory marker. And this marker of inflammation accounts for patients' IL-6, neutrophil, and tissue damage, which are kind of clinical indicators of hyperinflammation. And then what we did is we ordered patients along the x-axis according to their inflammation marker, according to this value. And we looked at comparing them to, for example, maximum IL-6 concentrations, so their maximum IL-6 peak value, their maximum T cell value, the maximum value of their inflammatory macrophages, and their the time to the interferon peak. And as you can see, there are some clear correlations and anti-correlations with these values. For example, those who have higher inflammation, so more severe disease, have fewer T cells, which is what we would expect. But in particular, what is interesting here is that there was a separation in time with the time to interferon peak. You see this kind of step process. So there's a clear kind of separation in this virtual cohort between those who had high inflammatory markers and therefore had more significant delayed peaks of interferon signaling, and those who did a good job of controlling the disease and had, really mild cases. And this, again, is an emergent property. This was something we didn't expect and was very interesting to find. And so we really wanted to kind of dig into that a bit further to try and understand, what could be regulating this kind of really what seems almost like an on-off switch in the interferon response. And again, here I'll just reiterate, even though I've said it several times, that this kind of delayed type 1 interferon response that correlates with disease severity is something that is really emerging in clinical cohorts now in COVID-19, but also was present or observed at least in SARS original strain and MERS. So what we did is we looked at plotting patients again by their inflammatory marker and then coloring these by the production rates of interferon and also the differentiation rate of monocytes, and then indicating, those with severe or those with mild disease. And what we found was that in the top here, T cell numbers were more strongly related to IL-6 concentrations than to interferon. And so even though this delayed interferon is important for distinguishing severe COVID-19, it's really the IL-6 that is driving that. This is interesting given that there's a negative feedback relationship for IL-6 and T cells in our model. But most importantly, what we found is in the bottom left, that the largest predictor of severe immunopathology and severe COVID-19 in our virtual cohort was linked to hampered monocyte-to-macrophage differentiation. Interestingly, in discussion with our clinical collaborators, and also this has been seen in other autoimmune and inflammatory diseases, and they're also seeing that there is some sort of dysregulation in the monocyte macrophage axis, and our model seems to be picking up on that. This is something that should potentially be probed further. Obviously, we can't, in a clinical setting, estimate differentiation rates, but there are exogenous cytokines that can control or help to orient this differentiation, and they may be potential therapeutic options for the treatment of COVID-19. We also found that in the severity was linked to the production rate of interferon, which might seem a little bit more obvious than this monocyte to macrophage differentiation, given that interferon is really a strong regulator of the severe COVID-19 response. So I'm just going to summarize for what we've done now before I kind of discuss some ongoing work that we've been doing. So just to summarize here, we've predicted that the delayed interferon signals are indicative of severe disease. And again, this is not something that is potentially surprising, I guess, but it is a kind of sanity check in that way. We found particularly that the impaired production of interferon and differentiation of monocytes into macrophages are correlated with severe COVID-19. And this is kind of the culmination of this portion of the work. You know, we aren't at this point looking even at treatments. We're really just trying to identify mechanisms of immunopathology. But of course, you know, we're still working with this model, we're still working to improve it, and we're going to use it for a number of different questions that are important not only within host, but also with regards to epidemic spread. So some of the questions that we're going to dig into and that I'm just going to talk a little bit about in the remaining time are, for example, how viral loads impact on disease severity. This isn't something I mentioned in what I've shown you, but we don't find necessarily a huge impact from heterogeneity in viral loads within the host on the immune pathology. So that doesn't necessarily mean that, you know, obviously higher viral loads, you might be more likely to transmit to someone else, but really we're just thinking of in host, it's more the immune response, the patient-specific immune response that's driving the severity of COVID-19 in our model than viral loads. And of course, because we've all been talking about new variants, we'd like to try and use this model similarly to what we did in the HIV case to understand how variants might emerge through selective pressure exerted by the immune system, despite the fact that SARS-CoV-2 is a slowly mutating virus. Our collaborators have picked up some in-host mutations. And we also, of course, want to understand what their impact is on COVID-19 severity. So in the last few minutes that are remaining, I'll just kind of talk about this, but this is unpublished and in its nascent state. So I'm happy to discuss more to kind of reorient this work. So I'm showing again our team. To this team, we add Julie Hussein, who's at the Montreal Heart Institute, and my new PhD student, Xiaoyan Deng. And this part of this work was particularly done by Viv Crow, who's just a super talented undergraduate at Concordia, who worked with Julie and I this summer to model kind of the emergence of genetic variation in the virus. So to start, before I get into the genetic evolution of the virus and how this impacts or can be taken into account in our modeling, the easiest target or question would be to look at how inoculation size, so the amount of virus that you're exposed to, affects disease severity in our virtual cohort. So for these simulations, we actually use the viral kinetics from the human data that I showed you earlier. So we expanded virtual patients based on the human viral kinetics. And we simulated each patient for 10 different inoculations, and they're quite small. In our initial story, so what I was telling you before, we used inoculation size of log 12 log copies per ml. And that's because it was based on the inoculation studies in macaques. And so now we expanded this. So we looked at higher inoculation sizes, so 14 log copies per ml, and then all the way down to 0.001 log copies per ml. And for each simulation, we measured 7 different metrics. Again, I know this is small, but I was trying to show it all in one slide. So the maximum interferon concentration, maximum IL-6 concentration, neutrophil concentration, T cell concentration, the time to neutrophil peak, the time to interferon peak, the maximum viral load, and the inflammatory marker. And this is kind of just a sanity check because this should be consistent across this cohort. And then what we did is we kept the order. We ordered patients by their inflammatory marker like we did before, and we kept the order between each of these characteristics. And so what we measured is the difference in the outcome relative to what we had seen in the base case at 12. So it's kind of, these are kind of hard heat maps to get your mind around. And I've had the same I have to keep reminding myself what these are. And we're working on thinking of other ways to kind of look at this. But for right now, this is what we've got. So the heat map is colored for their value relative to inoculation with 12. And what our results suggest is that the sensitivity of disease severity is primarily determined by the patient's propensity to having severe disease responses and not to viral load. And again, that's what we found in our initial simulations, but here we're really seeing it kind of more starkly. So really it's the patient-specific innate immune response that is driving disease severity, and it's not necessarily the exposure or the differences within the host of the viral load that we're seeing in our virtual patients driving disease severity. We also observed that patients that had severe disease responses in the base case for 12 log copies per ml, and those are patients 125 to 200, had little to no changes in their interferon, IL-6, or neutrophil dynamics. So really this also kind of supports this idea that the patients that have propensity towards severe disease have it because of some innate response within them. So that was, we're digging into that. We're trying, as I said, to think of an easier way to look at that. And we're also in parallel looking at the emergence of variants from in-host pressures. So of course, we're all familiar with the D614G mutation in the spike protein that emerged and became a dominant sequence early on, and also the viral variants like the so-called UK and South African and Brazil variants that may pose challenges to non-pharmaceutical and pharmaceutical interventions and potentially vaccine efforts, though it seems like this may not be as large a concern necessarily, especially with the mRNA vaccines. And so to kind of answer how these variants may emerge through within host selective pressures and also what their impact may be on disease severity, we integrate within host evolution of the virus from our Julie Hussain's team, who's focused on using population genetics. What they do is they use sequenced data from Gizaid and Biobank Quebec and analyze and determine single nucleotide substitution rates, track the emergence of mutants during infection, track the emergence of mutants, of course, across the globe. And so we account for mutations by calculating the probability, let me just go back here. So the idea is here that we have these characteristics in our patients, and now what we're adding in are different viral variants or mutations, and each of these mutations, they're arising based on measurements of how fast this is happening at a per nucleotide substitution rate. So we account for these mutations by calculating them, the probability of them arising at any position in the genome based on the sequencing analysis of Julie's team. And for example, what I'm showing here for these rates was after cleaning, we found something like 283,000 substitution events in 36,000 about samples. And what Viv did was to then construct a directed graph to find all the pathways between two different codons, and the probabilities for each of these pathways is then summed using the codon bias of the SARS-CoV-2 virus as weights. And so I'm happy to go into more details. I'm obviously going a bit quick here, but the idea is that we're using what we know about genetic variation within the viral sequence, genetic sequence, to be able to kind of predict how, at what rate there might be emergence of different variants. We started off maybe a bit simpler, I'd say, by imposing specific mutations. So we started by looking at the D614G mutation, the spike protein. Of course, we could use the other ones that are now more of concern and have arisen. But we also look at mutations in the ORF6 region. ORF6 and ORF8 have been shown to tamp down the type 1 interferon response. And so our question is kind of the opposite of the idea of where I was saying, you know, how does the immune pressure, how do these variants arise? And it's more, how would these kinds of mutations impact on severity, and what do we see in our patients? And so to do this, what we have is now 3 viral types with some probability of mutation between the viruses. So we have a wild type, a variant where there's a spike protein mutation, a variant where there's an ORF6 mutation. In the normal case, you have, exactly as I described earlier, susceptible cells, the whole model that we've been looking at, that become infected by wild type virus, and these infected cells secrete interferon. If you have a spike protein mutation, we model this as some modulation of the infectivity rate of this particular variant. And so there's some change in some increase of the rate at which cells become infected. And if you have an interferon mediating or interfering, which is a bad pun, protein or mutation, then there's no change in the infectivity, but there is a tamping down of the efficacy, essentially, of interferon signaling, and how, and then, you know, how the disease can continue, or the infection replication cycle continues. And so these are just really, again, preliminary results, but what we can do is perform parameter sweeps with respect to the same considerations that I showed you earlier for the inoculations, and we can look at how changes to these, to either the infectivity or the interferon response mediate the disease severity with respect to a variety of characteristics. And then, of course, you can think of this as a dual variant. So in some of these, you might have variants or mutations in the spike protein and elsewhere in other parts of the virus. And so this is something that we are looking at to understand how mutations impact on immunopathology in COVID-19. So I will stop there because that was a lot of information. And I know I went potentially a bit fast, but on stuff that we're still working from. But I'd like to just, again, really reiterate how interdisciplinary this project is and how collaborative it is. So just again, all of the people that have worked on this with me and with us, in particular, Courtney, Amber, Rosemary, Viv, Penny, Adrienne, and Sophia, who worked on the first part, and Julie and Viv and Xiaoyan that are now working with us on the second part. And of course, I'd like to thank our funders. And if you're interested, like I mentioned, the preprint is available online now. Please do check it out, and I'd be happy to take questions and to discuss further. So thanks very much.
Thank you, Morgan. And just a note that not only just interdisciplinary, I'm very impressed. I just surprised how many undergraduate students you can engage for this high-level training.
Yeah.
Now, with that, so it's open for questions. You can raise the questions directly, and you can also send by chat.
Maybe I'll start not with a question, but a comment. Morgan, that was really a clear talk. I know you crammed a lot of material there, but even for a person like me who's not in the field, I could see, I'll get a sense of what you were doing. It's very, very clear and very interesting.
Great. Well, thank you. I know it's always tough because we're so excited and we've been working on this day in, day out for a long time, but I'm glad that at least it came through.
So Morgan, I can ask a question. It's a question that I ask many people, and most of people criticize for too naive, not if they don't say it's too stupid. So how the repeated exposure within short period of time play a role in terms of the initial conditions? So
sorry, can you say it again?
The repeated exposure. Would that really increase?
Yeah. That is a really great question. We haven't looked into it, but we could by, simulating another, like we would simulate a drug, like a secondary peak, essentially. I'm not actually sure how that would change it. In some way, like I said, our model seems not impervious, but that really what's driving the response in the model is the individual characteristics of immune response as opposed to viral load. However, I haven't thought about repeated exposures and also the timing of those exposures with respect to if you're in this part of the curve or in this part of the curve,
exactly. Yeah.
how that's going to change. And that's something we should dig into. So that's a really great question. I'm not sure. That's my best answer.
Okay. It needs to be very polite. You're not saying the stupid question.
No, it's not actually. I think it's a great question. I hadn't thought of that. We've been so focused, on thinking of inoculation and variants, but we haven't really thought about that. But one of the things that Xiaoyan is going to do in her PhD is to look at within host and hopefully between host. And I think that becomes really an important question and something that we could dig into further. So that's I like the suggestion. Thank you.
Thank you. Other questions, comments?
Can you put your preprint link in the chat so we can click on it?
Let me see if I can do that. Sorry.
Looks like we need to invite you back very soon for the new variants. We're really making progress.
Oh, I think Jacques has a question.
Hi, Jacques.
Hi, Morgan. Yes, I have a question. What about the new variants?
Yes, so we haven't done them specifically, but we can. It's in the pipeline.
Okay. Do you have preliminary prejudices?
Well, so when we're looking at the D614G mutation in the spike protein, the way we model the spike protein, so yes, so we don't really see a lot of within host emergence of variants. So I think that this tracks well with the idea that these new variants came potentially from patients who were treated for a long time and who had a long infection course, because this infection course, even in our severe patients, it's not a huge amount of time, and we're not right now modeling treatment. So we see some rise in different variants within the patient, but we don't see extensive amounts of that. And so the way that we're modeling the spike protein mutation would be similar between the D614G and the new variants. So with respect to spike protein mutations, it doesn't seem to have a huge influence. However, the caveat being that, I don't know if this happens to you, but I had a late night research idea that hasn't been implemented yet, but that came to me in the middle of the night, which was really that we should be looking at thresholds beyond which this protein or this mutation could have a big impact. So we haven't looked at how far you need to push it. In the range that we've looked at it now, it doesn't seem to be hugely affecting the infection course, is what I could say.
Okay.
I want to continue Jock's question. So in terms of in-host models, so how the increase of the transmissibility is reflected in the in-host model?
Yeah, to be honest with you, I'm not, I know the epidemiological models, but I don't know as much the between host type stuff. There's some suggestion that obviously, as I said, that the increased viral loads for the immunopathology doesn't seem to have a huge effect, but increased viral loads will have a big effect on between host transmissibility. And so that's, I think, particularly where we would see a difference. We do see changes in viral load over the course of the infection with spike protein mutations, because there's a higher infectivity rate of within, like the cell-to-cell infection. And so those could increase transmissibility just through increased viral loads. But I guess also you could think of it on the flip side that if you have a variant, and this again is all speculation, but if you had a variant that is capable at the cellular level of being more infective, then if you transmit that, it might be just more easily transmitted because it's, you know, it's getting into cells a little bit easier. So I think that there's kind of like these dual processes that are happening in that respect.
Mm-hmm. Thank you. So it is the viral node decide on whether it's a mild or a severe case.
In our simulations, in our virtual cohort, we didn't find that viral load had a hugely impactful. It was hugely impactful on outcomes. What we really found was the intrinsic immune characteristics drove that response. So we had heterogeneity in our viral loads, but that didn't seem to really drive it, and that again was kind of mirrored in our inoculation simulations where we looked at changing the initial viral load. Which is kind of not great news, I guess, in some sense, because if that bears out, then that means that you get severe disease or you don't. It doesn't matter how much virus. I mean, I guess there is obviously going to be some influence of virus, but that people who have a propensity towards severe disease have that propensity with some degree, irrespective of viral loads.
Yeah, you have to somehow boost their native immunity or something.
Yeah, so like I said, if this monocyte macrophage axis is the one that seems to be suggestive in our predictions and also in some of the clinicians that we work with in their data, then, you know, trying to find treatment options that modulate this kind of, which is something, as I mentioned, in certain inflammatory responses has been, inflammatory diseases, excuse me, has been suggested before, may be quite a useful way to basically encourage this differentiation to happen.
If no other question, I have another question for you, Morgan, is how big the data you have? The most problem when I do the foodborne E. coli outbreak in-host modeling, my problem is I really don't have too many human data. I only have mouse. This is the same?
So we had, they're twofold. So in the data that we used to calibrate the model, there are human data in there because we used a lot of existing studies in different disease contexts or dose responses to do that calibration. But there are a number of in vitro and mouse studies in there, or other animal studies. We did our best wherever possible to get human or ex vivo studies so that we're doing, you know, as best we can. We do now have from hospitals in Montreal and places in the US, we do have a large amount of patient data, but we just got that. So, you know, part of the reasoning for mathematical modeling in some sense is to be able to do this a little bit faster, though I'm not so sure how much faster we did this than getting the data from clinical collaborators, given that ours just came out and they just gave me the data. So now we do have a large amount of complete blood counts and fax data from patients who are hospitalized and also from healthy controls.
Right. Great, thank you. I'm sure Kumar is happy to hear about this.
yeah, because we're talking about, I mean, if you can do simulations, I presume you can generate tons of data. No?
Yeah, I mean, that's actually something that we've been thinking about, not necessarily in this context, but we have this kind of platform where my lab deals a lot with heterogeneity. And so we can generate heterogeneous data, and we're semi-confident, you know, we are confident, as confident as you can be in a mathematical model. So then you could use this to generate synthetic data to use in, you know, deep learning or other processes, trajectory analysis. Exactly. So this is something that we've been thinking about with other people that we work with.
So maybe we should talk a little bit later.
Yes, please. Yeah, absolutely.
So I will take on the commission if you can develop some collective research. With this, other comments and questions? If not, again, I just want to, on behalf of the task force and the Fields Institute, I want to thank you, Morgan, for this very clear, interesting talk. It's very timely. And I just feel that we need to bring you back very soon. With that, again, thank you. And Kumar, is that okay that we conclude?
Yeah, good. See you all next week.
Yeah,
Bye.
thank you.
Thank you very much. Thanks
Bye.
for the invite.
Bye.
Bye.